My name is Luis Angel Rivera, and I'm going to be presenting you my work entitled Voluntary Exercise Reduces Colonic Reactive Gliosis in an Animal Model of Endometriosis. Our hypothesis is that endometriosis will induce colonic damage and reactive gliosis, which will be characterized by the increased expression of glial fibrillary acidic protein and S100B, which can be reversed by exposure to voluntary exercise. Our aim is to elucidate the impact endometriosis has on the enteric nervous system, which is known to be involved in pain development in other diseases, such as irritable bowel syndrome. Endometriosis is a chronic and complex gynecological disease that is characterized by the growth of endometrial-like tissue outside the uterine cavity, which results in pelvic pain, infertility, and gastrointestinal symptoms such as diarrhea, constipation, painful bowel movements, and bloating. It is interesting to note that gastrointestinal symptoms in endometriosis can be as common as gynecological symptoms, often leading to misdiagnosis with IBS. Recent studies have found that enteric glial cells are a major player in the abdominal pain found in IBS. The enteric glia are astrocytic-like cells that are localized in the enteric nervous system in the gastrointestinal tract. Normally, they express GFAP and S100B to maintain the gut barrier integrity and homeostasis, respectively. But when there is inflammation, the enteric glia shifts to a reactive phenotype, or reactive gliosis, which increases the expression of GFAP and secretion of S100B. The increased expression of S100B leads to oxidative stress and activation of NF-kappa B, driving inflammation, gut damage, and dysfunction. So, knowing that endometriosis causes gastrointestinal symptoms regardless of localization, we wanted to see if endometriosis induces changes to the enteric glia cells. So, for this study, we had 3 treatments. We had the sham that received sham surgery without implants. We had the endocontrol that received 4 autologous implants of uterus in the intestinal mesentery, and an endometriosis exercise group which received the implants and were exposed to voluntary exercise for 60 days after the endometriosis induction. On day 60, we sacrificed the animals assessed for endometriosis development colonic macroscopic score, which takes into consideration adhesions, ulceration, diarrhea, and thickness, and collected colon samples which were analyzed for GFAP and S100B using immunofluorescence. We took the pictures at 100x and analyzed the mean fluorescence intensity using ImageJ. All the statistics were done using GraphPath. First things first, it is important to note that all of the rats did exercise. Here you can see that the cumulative distance run by the individual animals per week. Although some ran more than others, they all did exercise. Here are example pictures of the immunofluorescence. In blue, we have DAPI staining the nuclei. In green, you can see GFAP, which gives the structure to the cell just like in astrocytes. And in red, we have S100B, which can be expressed in the nucleus and secreted. The final photo shows the merged image. Here, we found that endometriosis animals had significantly higher colon macroscopic scores than sham, and that exercise reduced the colonic damage. We also found that endometriosis increases significantly the expression of GFAP and slightly S100B, but exercise reduced this expression significantly in both cases. We also found in endo-exercise animals that the expression of S100B had a strong and significant negative correlation with cumulative distance run, where the more distance the rat ran, the less S100B expression was observed. Whereas GFAP expression showed a fair but not significant positive correlation with the macroscopic score, where the higher the macroscopic score, the more GFAP expression. The rest of the analysis didn't show any correlation whatsoever. In summary, endometriosis induces GI inflammation as noted by the increased colonic microscopy score and, and the increased expression of GFAP. Exposure to exercise reduced significantly the macroscopic score, GFAP expression, and S100p. Endometriosis animals exposed to exercise showed a strong and significant negative correlation between S100p and cumulative distance, while GFAP showed a tendency for a positive correlation with macroscopic score but did not reach significance. With these results, we conclude that endometriosis alters the normal enteric nervous system homeostasis, inducing reactive gliosis in the colon as noted by the increased expression of PFAP. The use of voluntary exercise appears to reestablish physiological homeostasis, reducing gastrointestinal inflammation in this animal model of endometriosis. And more importantly, this suggests that exercise as a complementary treatment for endometriosis could prove useful to reduce the burden of gastrointestinal symptoms that women with endometriosis experience. I would like to thank my thesis committee, especially my mentor, Dr. Appleyard, Dr. Noel, and Dr. Reveron, who have been of great help, the staff from my lab and BrainCore, and the funding that made this possible. Thank you very much for your attention, and these are the references. Thank you.